DNA, or deoxyribonucleic acid, contains all of the hereditary or genetic material in humans and most other organisms. So the DNA is what lets people know what genetic material is in a person or is in a certain creature or other organism. Now each cell in an organism has the same DNA. So if someone were to get a piece of your hair and a fingernail and a piece of skin and look at it very closely and look at the DNA in all three would be the same, and it would let whoever was looking at this know that all three of those articles came from the same person. This is the same for other organisms. Each cell in the organism's body is going to have the same DNA pattern so that you'll be able to tell that it came from the same creature. Most DNA is found in the nucleus of a cell. There is some DNA in the mitochondria, and then it is known as mitochondrial DNA, or it would look like this if you saw an abbreviation for it. So if you see the little MT before DNA in the abbreviation, that's because it's mitochondrial DNA and it's found in the mitochondria instead of the nucleus. But most DNA is going to be in the nucleus. Now, that important hereditary information found in DNA is stored as a code made up of 4 chemical bases. We've got adenine, guanine, cytosine, and thymine. And each one of those bases is only going to pair with one other base. They can't all pair up with any of the other 3. So adenine pairs with thymine, and guanine pairs with cytosine to form base pairs. And a way that I remember that is that the ones with the straight lines in their abbreviation letter, the A and T that can be made with straight lines, go together, and the ones that require a curved line go together. And that helps me remember that adenine pairs with thymine, these straight lines to form A and straight lines to form T, and guanine goes with cytosine because of the curved shape that it takes to make each of those abbreviation letters. The sequence of the adenine, thymine, guanine, cytosine pairs determines how the organism builds and maintains itself. So it's important to think about this. Not every organism or every person is going to have the exact same sequence of base pairs. And you might think of the alphabet. We use 26 letters to form all of our words and sentences, but the way that they're arranged, the sequence of those letters, is what lets us know what word is being communicated, what sentence is trying to be communicated. And each word has a different meaning, even though it's made up of the same 26 letters or some subset of those letters. So it works the same way. You're going to have some sequence of adenine, thymine, guanine, cytosine, and those base pair sequence is going to let you know, or let a scientist know, what that organism does to maintain itself, what kind it is. Is it functioning properly? Is everything in the right order? What does that order tell us? The same way that seeing CAT tells us that that is the word cat, and it's talking about a mammal with pointy ears and whiskers and a tail that's kept as a pet. Let's look at the next part. Each base pair, so our adenine-thymine pair or guanine-cytosine pair, is going to attach to a sugar molecule and to a phosphate molecule. And once those attach, once a base pair attaches to the sugar molecule and phosphate molecule, it forms a nucleotide. And these nucleotides form two long strands that spiral into a double helix. That is the shape that DNA takes on. And I did my best to give you all an example of a double helix. It kind of spirals around. If you were to curl a ribbon around my pen, it would form that kind of double helix shape we're talking about. And you should think of a twisted ladder with the base pairs as rungs. So if we were looking at this picture here, this would be our double helix shape that's spiraling around, and each one of these little rungs on the ladder would be a base pair. So we would have adenine and thymine together, and they would be paired up. And then here you would have guanine and cytosine paired up. And then you would have adenine and thymine, guanine and cytosine, all the way down the ladder, where you've got your sugar molecules and phosphate molecules that bound to your base pairs, making up the sides of the ladder here. And you just have this long DNA chain, and then the sequence that you see is going to let you know how that organism builds and maintains itself, because not every creature's DNA is going to be the same. Everyone has a very unique DNA pattern. DNA can replicate or make copies of itself by splitting the ladder in half. So if we're looking at this and we just decided to go through and cut this ladder in half, we would be separating those base pairs all the way down. So then you have AG, AG, and any other base pairs you've got. So we know we've got adenine, guanine, and because we know what's on one side of the ladder, each strand or half of that ladder serves as a pattern for duplicating bases. Since we know we've got adenine here, we know the thing that has to pair with it is thymine. If we've got guanine, cytosine has to pair with it. Since in DNA, adenine and thymine always pair together and guanine and cytosine pair together, you will know, or the cell will know, okay, I've got this DNA, I've only got half of it, but it's a pattern. If I have adenine, I need to add thymine to the other side. If I've got cytosine, I need to add guanine to the other side. So the cell will know, based on that half, that strand that it gets from DNA, how to make a complete DNA molecule and be able to form that double helix and complete the DNA strand. And each new cell needs an exact copy of DNA from the old cell. Because remember, each cell in an organism has the exact same DNA. So if the DNA is replicating itself, it needs to be giving an exact copy to a new cell so that this can continue, and the organism is going to continue to have the same DNA in it. So you will see a lot about DNA in biology, and it's important to remember that DNA is the hereditary or genetic material. It's usually found in the nucleus. And remember which bases pair with which. Your straight lines in adenine, A, pair with your straight lines in thymine, T, and your curve G for guanine goes with your curve C for cytosine. DNA is the hereditary or genetic material that is usually found in the nucleus, sometimes in the mitochondria. The most important thing to remember is that it's where all the genetic material is going to be found. It's a big identifier for a cell or for an organism. Hormones. A hormone is a chemical released by a cell, gland, or organ in one part of the body that affects cells in other parts of the body. It is basically a chemical messenger that transports a signal from one cell to another. So when your body needs something, a cell, gland, or organ that is a specialized one that can produce a hormone will release that hormone to go to another part of a body and tell it to do something else. So the hormones are there basically to keep your body in a state of equilibrium. So if it needs something in one place, then one part of your body may release hormones so that your body will react a certain way and basically get your body to where it needs to be to get that state of equilibrium. All multicellular organisms produce hormones. Even if they aren't doing the same things in every organism, more complex organisms are going to have more uses for hormones than simpler organisms are going to have. Here are some of the effects that hormones can have on a body. Stimulation or inhibition of growth. So hormones can tell your body, okay, it's time to grow a few inches. Okay, it's time to slow down and not grow anymore. Think about infants. Infants grow really, really quickly, and then they slow down as they become adults. And as you become an adult, you're not going to grow height-wise anymore, and your body is fully developed. It may still change, but the growth part, at least as far as getting taller and getting any bigger as a person, that is all done. Now, your body is still going to be telling your hormones will still tell your body to grow new skin cells and grow new other cells as needed. Hormones can affect mood swings. They can affect the induction or suppression of apoptosis, which is programmed cell death. So they can tell more cells to die, or tell your body to have less cells that are dying. So this is programmed cell death, and cells are supposed to die from time to time so that you can have new ones, but hormones can affect how many or few are dying at one time. Regulation of metabolism. Some people have faster metabolisms than others, and hormones have an effect on your metabolism. Activation or inhibition of the immune system. So your immune system, which is going to fight off bacteria or other foreign objects, how active or inactive it is can be affected by your hormones. Hunger cravings. Sometimes your body is going to have certain hunger cravings based on hormones that are going through your body. If you need a certain vitamin or mineral in your body, or a certain type of food, if you need protein, then your body may crave those things, and hormones can affect that. If you think about ladies that are pregnant having all those crazy hunger cravings, that's because they have a lot of different hormones going through their body at that particular time, and so they might crave something different, but usually it's based on what the baby or what they need. Preparation of the body for mating, fighting, fleeing, and other activities. If you've heard of the fight or flight instinct, this would mainly be the hormone adrenaline or epinephrine, where your body creates adrenaline and it gives you this powerful rush and you feel like you could do anything. So if you were getting ready to have to face something and fight or flight, or maybe before a big game, if you were nervous about the game and then you got really excited, you could have a lot of adrenaline. If you were going to go bungee jumping, anything that makes your body feel like you might be in danger or you might be about to experience something exciting, that could give you a rush of adrenaline. And so it prepares the body for that kind of a reaction. Preparation of the body for a new phase of life, such as puberty through teenage years, parenting, such as when a woman is going to become pregnant or becomes pregnant, and then we talked about the hunger cravings already, but the body is also going to grow and change a lot to accommodate the baby that's growing. And menopause, whenever a woman gets older and her body starts to change. Control of the reproductive cycle. So women have a monthly cycle, and that reproductive cycle is controlled by hormones. Sexual arousal, and the regulation of the production and release of other hormones. So certain hormones can tell your body to produce or release other hormones that are going to tell your body to do other things. So these hormones that would control the production and release of other hormones would be kind of like little manager hormones telling all the worker hormones to go out and do their jobs. And again, hormones control the internal environment of an organism through homeostasis, which just kind of means that the body is balanced. Everything in there is as it should be. Everything is at ideal levels, and that is what hormones are there to do. They're there to regulate things in the body and keep the body at equilibrium. kingdom Animalia. Animals have several characteristics that set them apart from other living things. Animals are eukaryotic and multicellular. So where other kingdoms can have creatures that are comprised of just one cell, animals are going to have more than one cell. They are also heterotrophic. This means they cannot create their own food and must rely on other sources of nourishment. So they're going to eat other plants or animals. They're not going to be able to create their own food with sunlight like plants can. Animals do not have cell walls, but they may have an exoskeleton, which is like a hard skeleton they secrete onto the outside of their body, or a shell. Now, they don't have to have that. Obviously, we don't have a hard exoskeleton or a shell, but some animals like your crawfish or lobsters would have an exoskeleton, or things like snails have a shell. All animals are motile. This means they can move, even if only during certain stages of life. So some animals may only move a little bit or very slowly, but all animals are motile, even if it's just for a short period of time or during one stage of their life. Nearly all animals undergo some form of sexual reproduction, but some animals are also capable of asexual reproduction. And this is possibly, and a lot of times, through parthenogenesis, which is when a fertilized egg is produced without any mating, budding, or fragmentation. So it is possible, but most of the time the animals are going to undergo sexual reproduction to produce offspring. Animalia is also known as metazoa, as far as a kingdom name goes, and it is divided into the subkingdoms Parazoa, which consist of sponges and trichoplax. Sponges, I'm sure you can think of, but trichoplax kind of looks like a puddle you spilled on the floor. It's very flat, but it is still an animal. It does have differentiated cells. It even has some eye cells on it. So Parazoa, such as the sponges and trichoplax, have differentiated cells but no distinct tissue or tissues or body symmetry. So they're kind of oddly shaped. Where most animals you would be able to see some symmetry, there is no symmetry as far as Parazoa are concerned. Now, all other animals are put into the subkingdom Eumetazoa. And these have 2 or 3 distinct layers of cells with differentiated tissues and either radial or bilateral symmetry. And what that means is that you can divide them a certain way. If they have radial symmetry, you would be able to divide them any way going in a circle, and the halves would match up. So a jellyfish would be an example of that. If you divided the jellyfish here, if you could divide it across here, if you could divide it across here, going any way in a circle around the top of the jellyfish, down across the top and down through all the tentacles, if you cut it, it would be more of a circular shape, and anywhere you cut it, it would be the same on either side. So it's symmetrical. And it would have radial symmetry because there isn't just one symmetrical line you can cut along. There's not just one line of symmetry, there are several. The same goes with a starfish. You could cut it along a line going this way, 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 or a line going this way. If you go all along that circle, it can have symmetry. It can have all these different lines of symmetry, and what's on the other side of the line of symmetry is going to match what's on the first side of the line of symmetry. Bilateral symmetry, you can only divide the body in half one way. So people have bilateral symmetry. You can divide straight down from the top of your head to between your 2 feet, and the 2 halves should match up pretty well, at least as far as body type and tissues are concerned. So people have bilateral symmetry. You can divide straight down the middle and the halves should match up pretty well. Butterflies or moths are another example that should match up. Cats, dogs, a lot of animals display bilateral body symmetry, but there are some that have radial symmetry. But these are all eumetazoa. Remember, the parazoa is not going to have any kind of body symmetry because it doesn't have distinct tissues and it is not going to have a distinct body shape every time. It's going to be different. So whenever you are thinking about kingdom Animalia, remember these certain characteristics that set them apart. They're eukaryotic and multicellular, made of more than one cell. They are heterotrophic. They cannot make their own food and must receive nourishment from other sources. They do not have cell walls, but may have an exoskeleton or a shell. And all animals are motile. They are able to move, at least at some stage in their life. Kingdom fungi. Fungi are a group of eukaryotic organisms. They used to be classified as plants, but genetic studies show that fungi are actually more similar to animals. Now, one reason that fungi were originally classified with plants is because they have a cell wall, just like plants do. But fungi have cell walls made of chitin, while plant cell walls are made of cellulose. So one of the differences that set fungi apart from plants is that their cell wall was composed of something completely different. Fungi can reproduce asexually via vegetative spores, or they can reproduce sexually via meiosis. A fruit body, such as a mushroom or mold, is formed where spores are created and released to begin new colonies. So when you think about a plant and how it produces a fruit, and that's where the seeds are that carries on life, it's kind of the same thing, except fungi and plants are not the same kingdom. So a fungi's fruit body is going to be in the form of a mushroom or mold, and instead of having seeds, it's going to produce spores that are released, and the spores go out and they create new colonies of fungi. Fungi include yeasts, molds, rusts, and familiar mushrooms. Something to remember is that slime molds and water molds, even though they are molds, are not grouped with true fungi. They used to be, but they are no longer considered true fungi because they just don't fit into this kingdom. So even if you see the word mold, that doesn't mean that it's definitely something that fits into kingdom fungi. Fungi grow in soil, on dead matter, and is symbionts of plants, animals, or other fungi. And being a symbiont means that a fungus can live on a plant, on an animal, or on some other fungus. So it can live on it. It could be a neutral relationship where that fungus lives there and it doesn't affect the plant, animal, or fungus that it's living on. It could be something positive where they're both getting something out of it, or it could be something negative where the fungus growing on that plant, animal, or other fungus is causing a problem. For instance, on certain rice plants, if a fungus grows on it, it can cause a disease that causes all those crops to go bad and not be good for consumption anymore. So it can be a bad relationship, or it can just be something neutral, or it can be something that's helpful for both the plant, animal, or fungus and the fungus that is growing on one of those things. And then a fungus doesn't always have to be a symbiont. It could just grow in soil or on dead matter all by itself. Fungi uses can include an essential role in decomposition. So when fungi grow on dead matter, they're going to help decompose that dead matter. It can be a direct source of food. Familiar mushrooms would be direct sources of food that you could eat, though not every mushroom, because fungi can be toxic. So don't think that every mushroom you see is all right or every fungus is going to be helpful. Fungi can be used as a leavening agent for bread. That's a yeast. Fermentation, such as in wine, beer, and soy sauce, and that would be yeast again. Antibiotics, enzymes and detergents, biological pesticides that could kill certain weeds, and then remember, fungi has a lot of uses, but it can be toxic. So, kingdom fungi is going to consist of eukaryotic organisms, and they're going to have cell walls made of chitin. They can reproduce asexually or sexually, and they are going to include yeast, molds, rust, and familiar mushrooms. Kingdom Plantae. Kingdom Plantae includes green algae, chlorophyta and charophyta, non-vascular plants such as liverworts, hornworts, and mosses, and non-vascular means they don't have any kind of veins to carry nutrients throughout the plant. Vascular plants, or tracheophytes, which means these do have veins that carry nutrients and water throughout the plant. And vascular plants include club mosses, ferns, horsetails, and seed plants, which are also known as spermophytes. And the seed plants include gymnosperms, such as the conifers, cycads, and the ginkgo tree, and these carry their seeds on the outside. The conifers would be like your spruce and pine trees, which have a cone, and that cone holds the seeds, and you can actually see the seeds coming out of it during the time of year where these seeds are coming out and the trees or plants are trying to reproduce. Angiosperms, or flowering plants, have their seeds on the inside in an ovary of the flower. And so both of those are seed plants, they just carry their seeds differently. The cell walls of plants consist primarily of cellulose and lignin. And lignin gives structure and strength to a plant cell wall, especially in trees, and it plays a large role in conducting water through the plant. Most plants engage in photosynthesis, which is where plants take in energy from the sun and use it to carry out its own processes. But some plants have lost that ability and must obtain their nourishment heterotrophically, so they can't make their own food. Autotrophs, such as those that carry out photosynthesis, are plants that can carry out photosynthesis. They can create their own food. But heterotrophs cannot create their own food, and they must get their nourishment from other places. So there are some plants that cannot carry out photosynthesis and must get their nourishment, their food sources, from other places. They can't make their own food. Green plants produce most of the world's molecular oxygen, and that would be done through photosynthesis. Plants can also serve as foods, such as grains, fruits, and vegetables, as ornaments rather. People have plants around their houses or in their offices that are just there for decoration. They're there as an ornament. They're not there for any specific purpose. And they've also been used as medicines or drugs. And there are some plants that can be used in a harmful way and a lot that can be used in a helpful way. And while you probably would go to the doctor if you needed a medicine for a long time, people didn't have doctors, and they used people that knew about herbs and plants, and those people would take care of them. And even once we had doctors would rely on a lot of plants, and a lot of your medicines that you would take over the counter now are created from plants or in a lab based on what the plant cellular makeup was. So plants play a large role in your medicines even today, even if you aren't getting them straight from some plant that is growing out of the ground. And the scientific study of plants is known as botany. So if you are interested in studying plants, you could become a botanist. There are lots of different categories in Kingdom Plantae, and the differences are based on if they are a green algae, if they have veins that help transport nutrients, and then you have different ones within those. So the vascular plants are the ones you're going to see the most of, and you will probably think most often of those that look like your flowering plants or your trees. Keep in mind that the cell walls of plants are made up of cellulose and lignin, where in fungi we had them made up of chitin. So that is a key difference between a plant and a fungus, is that the cell walls of plants are made of cellulose. Most plants are going to engage in photosynthesis, and plants have lots of uses, both as food or as an ornamental or decoration purpose, or for medicines. Meiosis. Sexual reproduction in eukaryotes involves a form of cell division known as meiosis. It has several different stages that are carried out to let one diploid germ cell result in 4 haploid gametes. And diploid means that it's going to have multiple copies of the genetic information, where haploid means it's going to have 1 copy. So, in this first part, this would be interphase, and it's kind of before meiosis starts, but it has to do with it because this diploid germ cell contains genetic information from both a maternal and paternal parents. So you've got female and male genetic material in this one diploid germ cell, and it's getting ready to divide to carry out sexual reproduction. So we have replication going on in here because each chromosome is going to double itself so that there's extra genetic material to carry out this process. In the next phase, we have prophase. And the replicated genetic material is going to line up in homologous pairs, which means that each one is the same genetic material. It's an exact copy. And they're going to cross over a little bit at these points here. And where those cross over, they kind of swap a little bit of genetic material. So each one of these pairs is going to take a little bit of the other pair, and that is called crossing over or recombination. So you're just kind of jumbling up the maternal and paternal bits before the division starts to take place. Then we move on to metaphase. And in metaphase, your centrosomes are starting to pull the homologous pairs apart. And then we move on to anaphase. And you can see they actually are getting pulled further apart. They're not crossing over anymore, and they're starting to move to opposite sides of the cell. And these are all prophase 1, metaphase 1, anaphase 1, because this is kind of a 2-step process. Then we keep going, and we've got telophase 1. In telophase 1, there's the cleavage furrow that's preparing for the cells to separate and become 2 distinct separate cells. There is a nucleus being reformed. The microtubules that have been connecting the chromosomes and pulling them apart have receded, and the genetic material is encased in a nucleus again. And then this last part is cytokinesis 1, where the cytoplasm is split, and there is a definite split made between those 2 cells. So now there are 2 diploid daughter cells. So we had our diploid germ cell the DNA, the genetic material was all replicated, so there was twice as much. We went through this cell division, so now each one of these daughter cells is a diploid daughter cell, and it has twice as much genetic material as it needs to be a haploid gamete. So we're going to go through what is known as meiosis 2, the second step in this process. So we have prophase 2. And in this phase, you'll see we've got the two daughter cells, and now they're kind of repeating the same process that happened up top. So there's no replication because that was an interphase, but they are going to start having the centrosomes move down to either side and start moving out to opposite poles of the cell and start pulling the chromosomes that way. In the next phase, we've got metaphase 2. You can see that the chromosomes have lined up along the equatorial plate in both of these daughter cells. The microtubules are connecting them to the centrosomes, and they're going to start pulling apart. So in our next phase, anaphase 2, you can see that actually happen. The chromosome pairs have gotten pulled apart, and now these individual chromatids are on separate sides of the cell, and they're still going to be kind of being reeled in by the microtubules at this point. And we move on to telophase 2, where we've got a nucleus forming around our new pairs of genetic material that have both the mother and father genetic material in each nucleus. They're all split up now, and we have the cleavage furrow again. The cells are getting ready to divide permanently. Your centrosomes have separated to the opposite sides still, and the microtubules have retracted. And then we go to cytokinesis 2, which is where the cytoplasm divided between these two new cells and these two new cells, and now we've got 4 haploid gametes, which means that they each just have one set of genetic information, and they're going to need a partner to be able to reproduce. So these gametes are going to be ready to be fertilized, and when they are fertilized, a diploid zygote will be formed. So these gametes are ready to be fertilized, and if they are, then they can go on to create a new eukaryotic organism. So meiosis does have a 2-part process, and it goes through the same phases as mitosis, but they're a little bit different because instead of just getting an exact copy, you're mixing genetic material from 2 different organisms, a maternal and paternal parent, and at the end you're going to have the 4 haploid gametes that each has genetic information from both parents. Mitosis. Eukaryotic cells reproduce asexually by dividing into 2 genetically identical daughter cells. Now they do this by the process of mitosis. So let's look. This first phase is called interphase, and it's not really considered a part of mitosis, but it's usually discussed with mitosis because it happens at the beginning right before mitosis happens. It's the build-up to mitosis. It's a period of cell growth and DNA replication. So the cell gets a little bigger, the nucleus gets a little bigger, the DNA replicates, so there's more of it for the mitosis that's about to happen. Chromosomes are in the form of uncondensed chromatin. And the nucleolus may still be present. So this would be our uncondensed chromatin. This darker dot here would be your nucleolus that you could still see. And these little pieces here would be your centrosomes. So whenever you see them along the rest of mitosis, remember these are the centrosomes. Okay, first stage of mitosis is going to be prophase. In prophase, the chromatin condenses into discrete chromosomes. So remember, it had gotten we had uncondensed chromatin that the DNA had been turned into. In prophase, it condenses into discrete chromosomes. So instead of a bunch of DNA squiggled up, you've got discrete chromosomes that kind of look like little squiggly X's. The nuclear membrane breaks down. So where we had a complete nuclear membrane here, it starts to break down a little bit. And sister chromatids are joined by centromeres. So what they mean by that is this would be your chromosome, and these sister chromosomes or chromatids are joined by these centromeres. So this would be your centromere. And then each of these would be sister chromatids that form the chromosome. The centrosomes are going to move to opposite poles of the cell. So remember, we just kind of had them in the cell here. Now they've moved to opposite sides of the cell, so they're opposite each other and they're kind of equally spaced in the cell. The chromosomes have paired up to form sister chromatids connected by a centromere. And now we're ready to move on to our next phase, which is metaphase. In metaphase, the chromosomes align at the equatorial plate. So where they're kind of just all over the place, they've lined up at the equatorial plate, which means that it's equally distanced from both of those centrosomes that have spaced themselves out at either pole. The microtubules attach sister chromosomes to the centrosomes. So you've got your centrosomes, and these are your microtubules that are attaching the chromosomes to the centrosomes. They've lined them all up along this equatorial plate. And now we're ready to move on to the next stage of mitosis, anaphase. In anaphase, the centromeres divide. So remember, your centromere was that little center here that was kind of holding the two chromatids together. So in anaphase, they divide. The microtubules shorten, so where they're longer, they shorten up, moving paired chromosomes to opposite sides of the cell. So we had these paired chromosomes, and now the microtubules are shortening. They're moving those individual chromosomes over closer to the centrosomes now. And then you're ready for the last phase of mitosis, which is telophase. In telophase, the cytoplasm divides along a cleavage furrow. And so you can see how it starts to kind of dip in. It cleaves in half, and so there's that cleavage furrow, and that's going to show you where your cytoplasm is going to eventually split completely off and form two new cells. The chromosomes condense to chromatin. Remember, that's what we had up here. We had all the squigglies. They're going to just start condensing to chromatin, not a discrete chromosome anymore. And the nucleolus reappears, and the nuclear membrane is going to be reforming around a clear nucleus. So you see the nuclear membrane is forming around where your condensing or your uncondensing chromosomes are, and your nucleolus has reappeared. So once these cells split off, then this is going to be a nucleus again that looks much like this, with the uncondensed chromatin and the nucleolus in the center. Two daughter cells, as we discussed at the beginning, two identical daughter cells will be formed once cytokinesis is complete. And cytokinesis is that process of the cell being cleaved in half to form two new identical daughter cells. So this is mitosis. It is how a eukaryotic cell can reproduce asexually. It just divides into two identical daughter cells. It goes through interphase, prophase, metaphase, anaphase, and telophase. So it starts off with this uncondensed DNA, this uncondensed chromatin genetic material. The chromosomes form little pairs, and the centrosomes come to either side. The centrosomes attach to the centromeres with microtubules, and those chromosomes line up along the equatorial plate. Then they split apart, starting to look like two new cells now, and each part of the chromosome pair goes toward a centrosome so that they are split equally across the cell. And then they split apart, forming a new nucleus with a nucleolus, and cytokinesis is going to be complete once this cell finishes cleaving in half. So just remember that eukaryotic cells can reproduce asexually by mitosis. Photosynthesis. Photosynthesis is a process used by plants and other autotrophic organisms to convert light energy, usually from the sun, into chemical energy that can be used by the organism. Photosynthesis usually gives off oxygen as a waste product as well. Oxygen is not something that the plant or other autotrophic organism will need in most cases, and so it will give off oxygen as a waste product. Now, autotrophic means that these organisms make their own food. They are capable of creating their own food. They can harness the light energy and create their own source of nourishment. Heterotrophs cannot do that and must rely on autotrophs or other heterotrophs for their nourishment. Most plants, most algae, and cyanobacteria perform photosynthesis and are called photoautotrophs. So, autotrophs are organisms that can create their own food. Photoautotrophs are organisms that create their own food from light, since photo translates to light, looking at the Latin root for that word. These are organisms that can create their own nourishment from light. Photosynthesis maintains atmospheric oxygen levels and supplies most of the energy necessary for life on Earth. So, as these autotrophic organisms take in carbon dioxide, they let out oxygen and help maintain the atmospheric oxygen levels, because people and other organisms exhale carbon dioxide, and the plants or other autotrophs take that carbon dioxide in and turn it back into oxygen, which we breathe. So if we didn't have photosynthesis, we'd be running low on oxygen. And so photosynthesis helps maintain our oxygen levels. It also supplies most of the energy necessary for life on Earth. Even though you don't feel like you're eating light or getting your energy just from light, if you eat a vegetable, then you're eating something that it has only been able to grow because it used light energy to provide nourishment for itself. So you are now eating that plant, which got its energy and was able to grow from light. If you were to eat a cow, if you had beef, if you had a hamburger, then you would be eating another heterotroph. Cows can't make their own food, but they ate grass and other vegetables and other foods that were grown, and they were grown by having light energy harnessed inside those plants. And so the cow got the energy from the plant, which got the energy from the light, and then you are getting energy from the cow. So it all comes back to the light energy. So while there are other sources of energy, the light energy is a primary source for everything going on Earth. Photosynthesis always begins when energy from light is absorbed by proteins that contain green chlorophyll pigments. So the autotrophs that perform photosynthesis don't always look green, but somewhere in their body, they're going to have these chlorophyll pigments that are able to take in and absorb light to start the process of photosynthesis. And photosynthesis occurs in two stages. You have your light reactions or light-dependent reactions, so these depend on light to happen. And it's when organisms capture and store the light energy. The light energy is converted to chemical energy in the form of NADPH and ATP. So in this light reaction or light-dependent reaction stage, the light energy is actually captured, turned into a chemical form of energy that can be stored or immediately used if necessary. In the carbon dioxide fixing reactions or light-independent reactions, these are ones that can happen even whenever light isn't present, as long as there was some energy harnessed in that plant already. So these don't have to happen while there is light present supplying energy. They can happen later. And this is when the formed NADPH and ATP, so it's already stored and formed in that autotroph, those two energy molecules drive the reduction of carbon dioxide to more useful organic compounds, such as glucose. And they are also referred to as the Calvin or Calvin-Benson cycle. So when the carbon dioxide is reduced, that process can be called the Calvin or Calvin-Benson cycle as well. So even though this is a light-independent reaction, it does still depend on molecules that were created by converting light energy into a stored chemical energy. So even though we call them light independent, they really couldn't occur if you didn't have the light transferring, the light energy that was able to be transferred to these energy molecules that are stored chemically. The main thing to remember about photosynthesis is that it is the process by which autotrophs can make their own energy using light. RNA, or ribonucleic acid, is made up of a base pair connected by sugar molecules and phosphate molecules. Now the bases in RNA are going to be adenine, uracil, guanine, and cytosine. Similar to DNA, they're going to pair with each other, and each one is only going to pair with one other base. They can't pair with any of the other three. So adenine pairs with uracil, and guanine pairs with cytosine. Guanine and cytosine also pair in DNA. Your curved letters for abbreviation pair together. And adenine and uracil pair together in RNA, and it's your vowels that are pairing together. Now in DNA, you have thymine as your other base, the base that pairs with adenine. But in RNA, you've got a different base. So you can think of it as your two abbreviations that are vowels paired together and your two curved abbreviations paired together. So adenine with uracil and guanine with cytosine. Usually, RNA is going to be single-stranded. But it can form a double helix, like the shape of DNA, by folding in on itself. So you have a single strand made up of sugar molecules and phosphate molecules, and then you've got the little base pairs coming off of it. So it may be like this. You've got your sugar and phosphate molecules, and then you've got your little base pairs coming off of it. And my little lines here are splitting it. So you would have adenine and uracil, guanine and cytosine, adenine and uracil, guanine and cytosine, all the way down. Now, if the RNA was more complex, it may fold in on itself, and this could curve around and kind of form a double helix by being tightly coiled and wrapped together, so that instead of having just one side of the ladder, it curved up and made the one side kind of sandwich it together and form into a double helix that way. But it's not going to be the same kind of pattern, the same structure as DNA is going to be. RNA is also a carrier of genetic material in some viruses. So, in viruses, RNA can carry the genetic material, and a lot of times, if it's a more complex virus, you'll have RNA that is a double helix. It has formed a more complex structure to carry genetic material for the viruses. In most cells, DNA is what's holding all of the genetic material, but RNA is what kind of moves it around the cell. So let's look at the three main types of RNA. Messenger RNA, or mRNA, carries codes from DNA in the nucleus to ribosomes in the cytoplasm. So it's moving, it's being a messenger and carrying that code from the DNA to ribosomes. Ribosomal RNA, or rRNA, they read that code carried by the messenger RNA. And ribosomes are composed of the ribosomal RNA and proteins. So the ribosomal RNA is a big part of ribosomes. And then transfer RNA, or tRNA, bring amino acids to the ribosomes where they are linked to proteins. So you can see RNA is moving around the cell a lot. It's not just staying in one place. DNA is mainly just going to stay in the nucleus and hold that genetic material. RNA is going to move it around, except in the case of viruses, where RNA is the main holder of genetic material instead of just a transport system. So let me show you what this might look like. So let's say we've got our ribosome, and so you've got RNA, your ribosomal RNA in here. Well, you've got DNA in the nucleus over here, and it's going to get transferred to RNA via the messenger RNA. So it's going to send that code along, and it's going to come into the ribosomal RNA. And then you've got transfer RNA out here, and it's going to bring amino acids into the ribosome. So you've got your ribosome here, and your ribosome holds the RNA. It takes in the mRNA, reads the code from DNA, it takes in the transfer RNA and picks up these amino acids, and out comes a protein. So the whole point of all this was to create a new protein, and RNA is a big part of protein synthesis. So it's going to move the code from the DNA via messenger RNA into a ribosome, and transfer RNA is going to bring amino acids into the ribosome, and the ribosomal RNA is going to take the code and take the amino acid and create a protein and spit it out. So that's what's going on here. Remember the big differences between RNA and DNA? DNA is holding all that information in the nucleus. RNA is moving the information around. It's how DNA gets its code from one place to another. So RNA is more of a transfer system within the cell, and it has several different kinds of RNA so that it can do different jobs in the cell, all working on transferring that genetic code. Viruses. Viruses are tiny, non-living, biological infectious agents. So while they are not technically alive, they are biological agents and they are infectious. They infect healthy cells. They are going to consist of either a DNA or RNA genome encased in a protein coat known as a capsid. So this capsid is protecting the viral genome until it can get where it needs to go to infect a host cell. And it can only reproduce, this is viruses, they can only reproduce by infecting and taking over a living host cell. So while they are non-living, they must take over a living host cell to be able to reproduce their viral DNA or RNA. Now, once they're inside a host cell, the virus may remain dormant for a while. So it doesn't always immediately enter the living host cell and attack. Sometimes it remains dormant for a while, but eventually it is stimulated into the active or lytic phase, and the virus inserts its own genetic material into that of the host. So the host cell is unaware of this, and the DNA that is, or RNA that is mixed in with the host DNA or RNA, it doesn't recognize that right away. So the virus is able to take control of the host's DNA by mixing with it and then taking over it. It causes the host cell to produce many viral genes and proteins. So once it gets in there, it starts saying reproduce, reproduce, make more genes and proteins. And the ones that it are making, the ones that this cell is making are going to be new viral proteins and genes instead of the normal cell's genes and proteins. And then all of these viral genes and proteins combine to form new virions or virus particles that destroy the host cell and are released to infect other cells. So what happens is, once the virus becomes active and it's in the lytic phase, it's going to insert its own genetic material into that of the host cell, so probably into the nucleus. The virus DNA or RNA is going to mix with the host DNA or RNA, take control of it, and then tell it to produce lots more of these viral genes and proteins, which combine to form new virions, and more are being made so fast that the cell can't keep up, and the cell is going to burst or lyse, which is where we get the lytic cycle or lytic phase from, because it is whenever the end result is going to be the cell lysing or bursting open. And once it bursts open, all these virions are released to infect other cells and go and repeat the process. Something to remember is that the genome of both DNA and RNA viruses can be either single or double-stranded. A lot of times a virus is going to not be too complex because the genome is only going to be 3 to 100 genes. It's not going to be very big, but it can be double-stranded if it is a more complex virus. So viruses are sneaky little things. They're going to be protected by their protein coat, the capsid, and insert their genome, their genetic material, into a host cell, take over, reproduce more viral genes and proteins, which form so many new virions that it causes the cell to burst open and send out more viruses into the host to attack other cells.
We're going to take a look at some parts of cell anatomy, starting with the nucleus, which contains the chromosomes and regulates the DNA of a cell. Now, the nucleus is responsible for passing on genetic traits between generations. The nucleus is a small structure and really is the defining structure of eukaryotic cells. And all eukaryotic cells have a nucleus. Now, chromosomes are highly condensed, thread-like rods of DNA. And DNA is the genetic material that stores information about the plant or animal. So since chromosomes contain DNA, then really chromosomes can contain or store the information about the plant or animal. Now we move on to chromatin, which consists of the DNA and the protein that makes up chromosomes. So there you can see a direct relation between chromosomes and chromatin. Now when we look at the nucleolus, this is involved in protein synthesis and synthesizes and stores RNA, which is ribonucleic acid. So the nucleolus is the structure contained within the nucleus, and so it consists of protein. It is small, round, doesn't have a membrane, but it's involved in what we know as protein synthesis and synthesizes and stores RNA. And so you notice here that the nucleolus, nuclear pores, nuclear envelope, and nucleoplasm all look very similar to the word nucleus. So these last four parts of cell anatomy that we're covering in this session all directly relate to the nucleus, because the nucleolus is contained within the nucleus. And then we have nuclear pores, which regulate the exchange of material that goes on in a cell. It regulates the exchange going on between the nucleus and the cytoplasm. Then we have the nuclear envelope, which consists of inner and outer membranes made of lipids, and it is basically a structure that encloses the structures of the nucleus. And then we have nucleoplasm, which if you're familiar with cytoplasm, it's very similar to that in the way it sounds, and that's because it's the liquid within the nucleus. And so it's very similar to cytoplasm. So that's a look at some of the parts of cell anatomy, especially those parts that relate directly to the nucleus. We're going to take a look at some parts of cell anatomy, starting with cytosol, which is liquid material in the cell. It is mostly water, but it also contains some floating molecules. And then we have cytoplasm, which is a term used to cover the cytosol and the substructures found within the plasma membrane. So the cytoplasm is a collective term for the cytosol, which is the liquid material in the cell, and the substructures, or we could say the organelles found within the plasma membrane. And now we come to the plasma membrane, which can also be called the cell membrane, which defines the cell by acting as a barrier. And so the cell membrane helps keep cytoplasm in and substances located outside the cell out. It also determines what is allowed to enter and exit the cell. And then we come to endoplasmic reticulum, which is the tubular network that comprises the transport system of a cell. Now there's two types of endoplasmic reticulum. The first is rough, meaning it has ribosomes on the surface, and smooth, meaning it does not have ribosomes on the surface. It is fused to the nuclear membrane and extends through the cytoplasm to the cell membrane. And then we have mitochondrion, which generates ATP and is involved in cell growth and death. Now, the cell structures vary in terms of size and quantities. So some cells may have one mitochondrion while others have thousands. And so mitochondrion performs various functions like generating ATP and being involved in cell growth and death. So that's a look at some parts of cell anatomy. We're going to take a look at some parts of cell anatomy, starting with ribosomes, which are involved in synthesizing proteins from amino acids. Now, ribosomes are numerous, making up about 1/4 of the cell. Some cells even contain thousands of ribosomes. Some of these ribosomes are mobile, and some are embedded in the rough endoplasmic reticulum. And then we have the Golgi complex, which can also be called the Golgi apparatus. And this is involved in synthesizing materials such as proteins that are transported out of the cell. Now, the Golgi complex is located near the nucleus and consists of layers of membranes. And then we have vacuoles, which are basically sacs used for things like storage, digestion, and waste removal. And then we have vesicles, which are small organelles within a cell. They have a membrane and perform varying functions, such as moving materials within a cell. And then finally we have the cytoskeleton, which consists of microtubules that help shape and support the cell. So that's a look at some parts of cell anatomy. We're going to take a look at some parts of cell anatomy, starting with microtubules, which are made of protein and are part of the cytoskeleton. And they also help support the cell. And then we have the centriole, which is involved in cellular division. Now, the centrioles are cylinder-shaped structures near the nucleus. Each cylinder consists of 9 groups of 3 microtubules, and the centrioles occur in pairs. And then we have the centrosome, which has a unique relationship to the centriole because it is comprised of the pair of centrioles located at right angles to each other and surrounded by protein. And so the centrosome is involved in mitosis and is also involved in the cell cycle. And then we have the lysosomes, which digest proteins, lipids, and carbohydrates, and it also transports undigested substances to the cell membrane so they can be removed. Now, the shape of a lysosome depends on the material being transported. So it digests some things, and things it can't digest, it transports. And then we have cilia, and the plural form of cilia is cilium. And so the cilium is appendages extending from the surface of the cell. And so these appendages cause the cell to move. And the movement of these appendages can also result in fluid being moved by the cell. So that's a look at some parts of cell anatomy. We're going to take a look at some parts of cell anatomy, starting with flagella. And flagella use whip-like movements to help the cell move. These are tail-like structures on cells, and so the flagella use a whip-like movement in order to propel the cell. Now these are similar to cilia, but are usually longer and not as numerous. A cell is generally only going to have one or a few flagella. Then we have the cell wall, which provides plants with a sturdy barrier. And this barrier can hold fluid within the cell. This cell wall is going to be made of cellulose and composed of numerous layers and surrounds the cell membrane. Then we have chloroplasts, which plant cells use for photosynthesis. Now, if you're not familiar with photosynthesis, this is the process plants use to create food energy from sunlight. Now, chloroplasts are specialized organelles, and chloroplasts also contain chlorophyll, which has a green color. Then we have plastids, which are used to make chemical compounds, and they're also used to store food. And so plastids are membrane-bound organelles found in plant cells. And plastids can also contain pigments used during photosynthesis. So plastids, like many of these other parts of a cell, can serve kind of double duty. They have a couple of functions that they contribute to. And then we have the plasmodesmata, which has a really long name, but this is the name for the channels between the cell walls of plant cells, and they allow for transport between cells. So that's a look at some parts of cell anatomy. A DNA mutation is when the normal gene sequence is altered. And gene disorders are the result of DNA mutations. So these DNA mutations cause gene disorders. Now, there's many ways DNA mutations can come about. So the first is hereditary. And this means the DNA mutation is passed from parent to child. So if a person has a gene disorder, it doesn't necessarily mean that their genes got messed up. It was just their parents' genes had a mutation. Now, you may ask, okay, well, how did their parents get that gene mutation? Well, it could have been passed down to them, or it could have been from one of these causes. One is called de novo, which means it occurs only in sex cells or shortly after fertilization. Then we have mosaicism, which occurs in a cell during an early embryonic stage. Now, the result of that is that certain cells are going to have this disorder, whereas you're not going to see the results of the mutation in other cells. And then we have acquired mutations, or we could call them somatic mutations. This is due to environmental factors or replication errors. Now, when I talk about replication errors, this means maybe an error that happened due to an incorrect base pairing. Or another way mutations can occur is if an error is made by the DNA polymerase while replicating a base. This happens about once for every 100 million bases. But a repair protein proofreads the code, and so the mistake is usually repaired. Now, there's a term called polymorphism, which means that the differences in humans, such as eye and hair color, may have originally been the result of gene mutations, but are now part of the normal variation of the species. So while these DNA mutations may seem like they're all bad. Sometimes they're somewhat neutral and don't really cause a problem, and now they're just a normal part of the variation of a species. Pairs of chromosomes are composed of DNA, which is tightly wound to conserve space. Now, when replication starts, the once tightly wound DNA unwinds. And so the steps in DNA replication are controlled by enzymes. Now, one of these enzymes is called helicase. The enzyme helicase instigates the deforming of hydrogen bonds between bases to split the two strands. Now, there's a couple of terms that you should be familiar with. One is the origin of replication. This is where the splitting starts. So remember, a minute ago, I said that the deforming of hydrogen bonds between bases leads to the splitting of the two strands of DNA. So the origin of replication is wherever the splitting of the two strands begins. And then we have the replication fork. This is the portion of the DNA that is unwound to be replicated. So in the process of DNA replication, each strand of DNA is transcribed by an mRNA. And so the M in mRNA stands for messenger. So this is messenger RNA. And so the mRNA copies the DNA onto itself base by base in a complementary manner. Now the exception here is that thymine is replaced by uracil. Now thymine is only in DNA, so it can't be transcribed over to RNA. And so thymine is replaced by uracil, which is only in RNA. So that's a look at the process of DNA replication.
The brain is one of the most important parts of our nervous system, and today we're going to go over the various parts of the brain and talk about some of the ways they contribute and help in the areas over which that part of our brain control. And so we're going to begin then with the frontal lobe. The frontal lobe controls our emotions, our judgments, and our motor aspects of our speech, and is also the primary motor cortex for voluntary muscle activation. After the frontal lobe, we have the parietal lobe, which receives sensory information from touch, proprioception, which has to do with our ability to tell where our arms and legs are and our orientation in space. So even with your eyes closed, you know where your arms and legs are. You have a sense of your body. This is proprioception. The parietal lobe helps govern that. Also our sensation of temperature and pain. That's the parietal lobe. Then the temporal lobe is responsible for our hearing, auditory information, and our language comprehension. The occipital lobe is our center for visual information, our sight. The cerebellum coordinates all of our muscle function. And then we have the brainstem, and within the brainstem we've got the midbrain, the pons, and the medulla. The brainstem governs our breathing, our respiration, our hearts, our cardiac center, and all the nerve pathways to the brain come through the brain stem. And then finally, moving on then from that to the diencephalon, which is the thalamus, subthalamus, and hypothalamus. The thalamus helps integrate and relay sensory information from the face, feelings there, our retinas, our cochlea from our ears, our taste receptors, and interprets sensation of touch, pain, and temperature. So our thalamus is very important to not only our sense of what we feel in terms of pain, temperature, touch, but also our retinas, our eyes, our cochlea, and our ears, and our taste and our tongue. The hypothalamus controls the autonomic nervous system and the neuroendocrine system. It maintains our body homeostasis, helps regulate body temperature, helps regulate our appetites, governs our thirst and how we feel that, our sleeping cycle, and hormone secretions. So once again, the brain very integral, necessary, core part of our nervous system, broken up into these various regions and what they controlled. If you'd like to find out more about the brain and its relation to our nervous system, I encourage you to go to the link that you'll find below this video. At that website, you'll find an ebook that is ready for immediate download.
Water is essential to life as we know it, and it is one of the main constituent of many living things. And so there's several interesting properties of water I want to take a look at. So first is its high polarity. It has negative and positive ends. And this is due to the fact that water is made up of 2 hydrogen atoms and only 1 oxygen atom. And also hydrogen bonding. Water molecules engage in hydrogen bonds with other water molecules. Water also is cohesive. And so cohesiveness means it's attracted to itself. You may have noticed this if you ever filled a cup of water up a little bit too high above the rim of the glass. Well, the water didn't immediately spill over because of cohesiveness. The water molecules were attracted to the other water molecules, so the water didn't necessarily spill out because the attraction was stronger than the force of gravity trying to pull it out. Then we also have adhesiveness, which means water molecules are attracted to other molecules. Water has a high specific heat, which means it resists the breaking of its hydrogen bonds and resists heat and motion. So specific heat means that it has a large amount of heat required to raise temperature by a specific amount. This also kind of goes along with high latent heat, which is the net heat change in change of state. So basically when water changes from one state to another, that means there was a lot of heat either absorbed or put off. And so net heat change means how much water was absorbed or exerted to do this change of state. Now, water in its solid form as ice floats in its liquid state of water. Now this is pretty odd because many times substances are denser in their solid forms, but water is strange in that its solid state floats in its liquid state. This is because when water is frozen, it expands, and so it becomes less dense because it's taking up a larger amount of space. So that same material is spread out over a larger amount of space, which means it's less dense than the liquid water. And so when anything is less dense than water, then it's going to float in that water. So that's why ice cubes float. So that's a look at some of the interesting properties of water. The functions of plant and animal cells vary greatly, and the functions of different cells within a single organism can also be vastly different. So we're going to take a look at plant and animal cells and the similarities and differences between them. And so first, let's take a look at the similarities. Plant and animal cells are both eukaryotic. So they're both eukaryotic as opposed to being prokaryotic. And since they're eukaryotic, they both have a nucleus. And then a third important similarity is their reproduction process. Both plant and animal cells reproduce by duplicating genetic material, separating it, and then dividing it in half. There are also some other similarities related to parts of the cells. They both have cell membranes, cytoplasm, microtubules as part of their cytoskeleton, vacuoles, and then many other structures are the same. Now they both have differences with each other. And so we have plant cells and animal cells. And so one huge difference is that plant cells have a cell wall. Animal cells have no cell wall. And so plant cells have a cell wall made of cellulose that can handle high levels of pressure within the cell, which can occur when liquid enters a plant cell. Now, plant cells have chloroplast. And these chloroplasts come in handy in a process known as photosynthesis. So plant cells have chloroplasts that are used during the process of photosynthesis, which is the conversion of sunlight into food. Chloroplasts in plants that perform photosynthesis absorb sunlight and then convert that into energy. Now, animal cells use something known as mitochondria. And so the mitochondria produce energy from food in animal cells. Another difference is that plant cells have a regular shape. In other words, they pretty much always have the same shape, while animal cells have many shapes. There are many possible shapes that an animal cell could have. And there's also a difference in size. Plant cells are larger than animal cells generally. And then finally, plant cells have cell plates, and so two cells are separated by cell plates, while animal cells pinch in half. So what that means is that plant cells build a cell plate between two new cells, while animal cells make a cleavage furrow and pinch in half. So that's not all the differences between these two types of cells, but these are many of the main differences, and these are many of the main similarities. A covalent bond is formed between two or more atoms that share pairs of valence electrons. Now, a single covalent bond is just one pair of shared electrons. Water, which is H2O, has two hydrogens to every one oxygen. Each hydrogen atom requires one valence electron to fill its outer shell, and oxygen requires two valence electrons to fill its outer shell. So, by sharing one valence electron with each hydrogen atom, oxygen fills its outer shell. Likewise, by sharing their valence electrons with oxygen, each hydrogen atom fills its outer shell. So, in water, oxygen has two covalent bonds, one to each hydrogen atom. So this looks something like this. You have oxygen with its inner shell, and then it also has an outer shell. So you have two electrons in its inner shell, and then oxygen has 6 total electrons in its outer shell. But remember, it needs 2 more valence electrons to fill its outer shell. And hydrogen looks something like this with 1 electron, and there's 2 hydrogen atoms just like that with 1 electron that need 1 more valence electron to fill their outer shell. So what happens here in covalent bonds is hydrogen comes over here and donates its valence electron to oxygen. Oxygen donates one of its valence electrons to hydrogen. So basically they're sharing these valence electrons. And same thing happens with this other hydrogen atom. And due to my drawing inabilities, this doesn't look very good, but these still are circles over here. And what's happening is hydrogen and oxygen are sharing these valence electrons so that both of their outer shells are filled. And then you have 2 covalent bonds here. You have one here and one here because a covalent bond is 1 pair of shared electrons. And there's a pair of shared electrons here and a pair of shared electrons there. Now I want to go over some things that are generally true about covalent bonds, but I'm not going to present these as facts because they're not facts and they're not laws. They're not always true. They're just generally true. And so covalent bonds generally form between nonmetals. And if you look to our example, here we have 2 covalent bonds formed between nonmetals because oxygen and hydrogen are both nonmetals. And then covalent bonds generally have lower bond energies than ionic bonds, which a bond energy is the amount of energy it takes to break a bond apart, which the energy is usually in the form of heat. And then the atoms of similar electronegativity value usually form covalent bonds. So the atoms that make covalent bonds up have similar electronegativity values. So those are some general facts about covalent bonds. If you just remember a couple of things from this session, remember that covalent bonds are formed between two or more atoms that share pairs of valence electrons. And a single covalent bond is just one pair of shared electrons. Ionic bonds tend to form under two certain criteria. And the first is ionic bonds tend to form between metals and nonmetals. So if you have two elements and one's a metal and one's a nonmetal, an ionic bond is likely to form. Now the second criteria is that ionic bonds tend to form between elements with a large difference in electronegativity. So the larger the difference in electronegativity, the better the chance ionic bonds are going to form. And so if elements meet both of these criteria, there's a very good chance they are very likely to form ionic bonds. Now, keep in mind that elements with the highest electronegativity values are in the upper right-hand corner of the periodic table of elements, while those with the lowest are near the lower left-hand corner. So you have elements with low electronegativity down here and elements with high electronegativity up here. And so just by knowing those two facts right there, if you see an element on the periodic table, if you just imagine this board as the periodic table, and it's about right here, you just know by where it's geographically situated on the board that there's a really good chance that it has a high electronegativity because it's near the top right-hand corner of the periodic table. Now, I have some pairs of elements here, and I want to examine these elements to see if they meet this criteria and determine whether or not they are likely to form ionic bonds. So first we have nitrogen and oxygen. Now, nitrogen is a nonmetal, and then oxygen is obviously a nonmetal. So they are not likely to form ionic bonds because there is not between a metal and a nonmetal. We have two nonmetals here. Then we have potassium and fluorine. And potassium is an alkali metal. And fluorine is a nonmetal. So we've met the first criteria. We have reaction here between a metal and a nonmetal. So we're doing good so far. And then there's a high electronegativity difference here. And it's actually 3.2, which is pretty high. So they're likely to form ionic bonds, specifically potassium chloride. Now here we have barium and sulfur. And barium is a metal. And then sulfur is a nonmetal. So here we have a metal, a nonmetal. We're doing good so far, so they're likely to form ionic bonds, specifically barium sulfide. And then in addition to that, their electronegativity difference is moderately large at about 1.6. Now finally, we have cesium and tin. And cesium is an alkali metal. And then I'm sure you can guess that tin is a metal. So here we have two metals interacting with each other, so it's not between a metal and a nonmetal. So these cesium and tin is not likely to form ionic bonds. So notice here the main factor here was that we had two nonmetals and two metals here. But when we have a metal and a nonmetal, they're likely to form ionic bonds, and especially if they have a high electronegativity difference. So hopefully through this short session, you have a better understanding of ionic bonds. The three laws of thermodynamics are important to our understanding of energy and heat. So I've written each law up here on the board, and then under each law, written a brief summary of that law. So the first law of thermodynamics says that energy is always conserved. So take, for example, when I rub my two hands together. So I'm rubbing my hands together. So I'm exerting energy to rub my hands together. So it would look like my body is losing energy because I'm having to exert that energy to rub my hands together. But because I'm rubbing my hands together, I'm creating friction, which creates heat, and I can feel my hands getting warm right now. So although energy is coming out of my body to rub my hands together, it's changing forms into the form of heat. So heat is another form of energy. So energy is always conserved. It may change forms, but the energy is always there. You can't destroy energy. The second law of thermodynamics is that a system will develop a uniform temperature. So say I had a box, and inside that box I had 2 blocks of metal, and the blocks of metal were of equal weight. The first block of metal weighs 100 degrees, and the second block of metal weighs, the first block of metal is 100 degrees, and the second block of metal is 0 degrees. So eventually those 2 blocks are going to balance each other out until both blocks weigh 50 degrees. So that's basically the second law of thermodynamics, that both blocks in the box are going to work together to find one temperature, an equilibrium, a temperature that each block is going to have. So this can be applied to many different things, any sort of system that you have where you have different objects inside that system and they have different temperatures. Eventually all the objects in that system are going to have the same temperature. The third law of thermodynamics is that entropy becomes constant as temperature approaches 0. Now this doesn't really make sense unless you know what entropy is. Entropy is the flow of heat. So as the temperature approaches 0, the flow of heat becomes constant. So that's a look at the 3 laws of thermodynamics and how they affect our world. In metal cations are surrounded by a sea of valence electrons. And these valence electrons are said to be delocalized. So I'm going to write that up here on the board because that's an important point. And what delocalized means is that these valence electrons aren't attached to a single atom. Instead, they float around many atoms. However, the non-valence electrons are still associated with individual atoms in the metal. All right, so in addition to being delocalized, valence electrons are attracted to metal cations, and that's what creates the metallic bond. So I'm going to write up here on the board, attracted to metal cations. So I hope you're tracking with me. Remember, valence electrons are delocalized. They're attracted to metal cations, creating metallic bonds. Now, because these valence electrons are not held tightly in the metals, they have different properties from other ionic solids. So this means they are malleable, which means they're able to bend. They are also ductile. They can be drawn into wire. So in addition to being malleable and ductile, metals have varying melting points from low to high. And the sea of electrons moves freely and conducts electricity, so metals are good conductors. So I'm going to write good conductors. We also said they have variable melting points. So the important thing to remember is that metal cations are surrounded by a sea of valence electrons that aren't attached to any one atom, but just float around. And they're attracted to the metal cations, forming metallic bonds. And because these valence electrons are not held tightly in metals, they have different properties from ionic solids. We can classify cells into 2 categories, prokaryotic and eukaryotic. So prokaryotic cells are smaller than eukaryotic cells. In addition to being smaller, they lack membrane-bound nucleuses. So they don't have a membrane-bound nucleus. Now, they contain single-stranded circular DNA molecules that free float in, you can probably guess, they free float in cytoplasm, because cytoplasm is kind of the fluid in a cell that all the other organelles in the cell float around in. So the DNA molecules free float in cytoplasm. So some different organelles that are in prokaryotic cells are ribosomes, centrioles, often one or more flagella, and cytoskeleton elements. Then we have eukaryotic cells, and so these are going to be obviously larger than prokaryotic cells. And these contain linear, double-stranded DNA combined with histone and packaged as chromosomes. And then the chromosomes are found inside a nucleus bounded by two membranes. So notice here, the eukaryotic cells do have a membrane-bound nucleus. Now, eukaryotic cells contain a variety of other membrane-bound organelles, such as mitochondria, chloroplast, endoplasmic reticula, Golgi bodies, lysosomes, and vacuoles. Now, eukaryotic ribosomes are larger and more complex than those found in prokaryotes, which is what is another way of saying prokaryotic cells. Now, you may be wondering, where do you find prokaryotic cells and where do you find eukaryotic cells? Well, prokaryotic cells are usually in single-celled organisms, such as bacteria, whereas eukaryotic cells are what you find in more complex organisms. It's what you see the result of on a daily basis, because plants and animals are made up of eukaryotic cells. So that's a look at prokaryotic and eukaryotic cells and the differences between them.
Sickle cell disease is one of the most common genetic diseases in the United States, typically among African, Middle Eastern, Mediterranean, and Indian descent. And sickle cell disease is caused by an abnormal globulin gene that causes the hemoglobin S to form a sickle shape rather than a round shape. Now, the abnormal hemoglobin shortens the life of the hemoglobin, so it causes a chronic anemic state. And the crisis occurs when these sickle cells clump together, and it can cause thrombi, vascular occlusions, hypoxia, and even a myocardial infarction. Now, some of the most common symptoms of sickle cell disease are pallor, jaundice, weakness, and fatigue. Also, some people experience pain with sickle cell disease, and it can vary in severity and also frequency. Now, some of the presenting signs may be pale lips, tongue, palms, or nail beds. There may also be lethargy, difficulty waking, listlessness, irritability, or fever. And people with sickle cell disease are typically more at risk for bacterial infections. Also, children are generally hospitalized less than 6 times a year. And adults who are hospitalized more than 3 times a year, that may indicate impending death.